So, as we all know, the Hope Foundation has been very generous and very instrumental in funding projects which can impact our clinical trials. And the next is a presentation from Omar Aljitawi from the University of Kansas, where is a very interesting protocol that was funded by the Hope Foundation, and we hope that we will have more similar to this in the future. So this is a great example of translational science going straight to the clinic.
Good afternoon. Thank you, Dr. Ellis, for this invitation and nice introduction. I'm very excited to be here today to present this concept. And I want to say there's nothing for me to disclose except that HBO does not refer to the cable channel HBO, so it's totally different. I'm going to explain as we go, okay? So what is the problem that we're trying to fix here today? Well, actually, I've been working on it for a few years now. So if you look at hematopoietic stem cell transplantation, which cures hematologic malignancies, clinically, what we do is we submit patients to conditioning regimen, which is typically chemotherapy and radiation. Then we infuse the hematopoietic stem cells, and afterwards, these patients, because of the effects of chemo and radiation, go through a period of neutropenia where their counts are low, they're susceptible to having infections. And what happens is after the stem cells do their job, the patients engraft, and basically they recover their neutropenia. What happened is, in the area that I'm interested in, which is umbilical cord blood transplantation, we have issues here because neutropenia is usually prolonged because of delayed engraftment. Now, this is clinically. So what happens actually behind the scenes biologically? What happens really is that when we infuse the cells into the blood, we expect them to home to the bone marrow, and then after that, we expect them to engraft. And this is actually where the problem in cord blood transplantation, mainly because of the limited number of stem cells in the cord units, but also because of impaired homing to the bone marrow. So this is the piece of the puzzle that we're trying to fix here. So for those who don't know much about transplant, let me introduce you to the idea of homing. What happens is when patients get transplanted, we infuse the cells into the blood, and what we hope for them to do is move from the vascular space into the bone marrow space and create niches there and populate and then produce cells that end up going to the bone marrow, to the blood, which is what we call engraftment. This actually is driven by attraction to an SDF1, which is a chemokine in the bone marrow. Now, what I want you to know that this process is so transient. It actually happens in the first 72 hours after you infuse the stem cells. So it's a very limited window to intervene. I always been intrigued by this observation, why cord blood transplants, cord blood stem cells have problem with homing to the bone marrow. And one day I was reading this paper, and that was kind of the eureka moment in my career. What happened is, I'm going to walk you through it carefully here. What they did, they looked at the hematopoietic, or I want to say CD34 positive cells in the peripheral blood of newborn babies. And CD34, as you know, is a marker of stem cells, and they show here that there are 25% initially, and over a few hours, they go to, actually over hours, it go down to 5%. They say, because they looked at many markers of bone marrow homing, they speculate that what happens is that stem cells end up homing to the bone marrow. That's why they clear from peripheral blood. What was interesting is they show on a separate curve that the hormone erythropoietin goes down over time. And if you look at the APO curve and the hematopoietic stem cell curve, you can see that they're kind of mirror image of each other. And that was kind of the moment that I was like, aha, I think I know what happened. What happened is, my prediction was, maybe because when the baby is born, lungs expand with oxygen, EPO levels go down, maybe that's actually the marker for stem cells to home to the bone marrow. And that's what led me to think there might be a link between erythropoietin and hematopoietic stem cell homing. So I developed this hypothesis at the point, and this figure is 10 years old now. And what happened is, I thought and this is based on several observations. I'm not going to talk about them today, but maybe low EPO enhances the homing process, while the opposite, which is high EPO, enhances mobilization out of the bone marrow. The question was, though, how can I lower EPO? And I did start PubMed search the same day I read the first paper and came up with this procedure, HBO. And I still remember it was Friday afternoon. I got out of the office, I went to the hallway, talked to my colleague, I said, I have a problem that I could fix. And I told them about the fix. And I was very excited about it. So today I'm going to go over the fix that I have. So HBO is not the cable channel. It's actually hyperbaric oxygen. I don't know if you know much about that, so I'm going to introduce it to you. Basically, it's when you breathe 100% oxygen under pressure. We breathe under 1 atmospheric pressure, so we go to like 2.5 atmospheric pressure. There are two things that we introduce, pressure, and also we introduce physiologic change, which is hyperoxia. So there are two interventions here. And this happens in a chamber that looks like that. I don't know if anyone can recognize the person in the chamber here. When I did a Google search for hyperbaric chambers, this picture came out. This is actually Michael Jackson. He used actually to use hyperbaric oxygen chambers to help him sleep until he got on the habit of using propofol. And what happened is, if you think about it, there was one paper that actually reported that in healthy volunteers, the use of hyperbaric oxygen can lower EPO. So that was kind of what introduced me to the idea of using hyperbaric oxygen. So in the next few years after that, we kind of embarked on multiple experiments to kind of study the effect of hyperbaric oxygen on EPO and engraftment in a murine model of transplant. I was totally foreign to this, but with the help of many people, we were able to do these experiments. We were able to show that when mice get exposed to hyperbaric oxygen, EPO levels were significantly lower. And also, they seem to retain more CD34 positive cells, or the stem cells, home better to the bone marrow of these mice. And over time, they have better engraftment. So we were very excited about these results. And because of the safety of the procedure, the availability, we have an access to one in our hospital. We decided to move it from the bench to the bedside. Initially, I want to say this. Initially, I thought of doing this as a clinical experiment, but ended up getting funding, but I was asked to do the mice work first. So this is actually the first design that I thought of doing. And what we did is, patients initially receive preparative regimen, again, which is chemotherapy or radiation. Day 0, which is by convention, is the day we infuse the stem cells. Patients early on get hyperbaric oxygen treatment, and we wait 6 hours from the time we start, or 4 hours from the end, and we infuse the cells. And the reason for that is because in the healthy volunteer study, that did show that exposure to hyperbaric oxygen results in lower EPO in 4 to 8 hours. So we decided to do it that way. And basically after that, we followed these patients, mainly for assessment and homing. I just want to say, when I present this concept and talk to people about it, they always think that I treat the unit. I'm not. I'm treating the patient. So what I thought is I'll actually introduce this picture because it's a picture of a patient who was actually treated on this study. And clearly she was very happy. What happened is, let's start with the results of the first trial, which is a pilot study in cord blood transplantation. The first concept here is, does hyperbaric oxygen lower EPO? And we did see in patients who received transplantation and hyperbaric oxygen, they actually did have lower EPO levels. The lowest EPO level was at 8 hours after hyperbaric oxygen. What's really interesting is, as you can see, there was a rebound in EPO that started 24 hours after the hyperbaric oxygen treatment. Just remember that. So, okay, we lowered the EPO. Does that translate into better engraftment? And here I show our data compared to our historic data. And the numbers are small because that was the first interim look at the data. But we did see that the median time to neutrophil recovery was shorter in patients who had the hyperbaric oxygen compared to our historic data. And the same thing for platelet count recovery. So after we had these good results with hyperbaric transplantation in cord blood, with hyperbaric oxygen in cord blood transplantation, we started asking the question, can this apply to other types of transplantation? One of them is autologous transplantation, which is when you use your own stem cells. And the goal was, again, we're going to study the safety. This is a newer setting. But also we're going to look at efficacy. Are we going to be able to shorten the period of neutropenia and thrombocytopenia, which will translate into improvement in transplant outcome, less morbidity, potentially less mortality, maybe saving, cost saving too. So we treated 20 patients to date. All of them tolerated the therapy except for one, and that patient had ear problems. He could not tolerate it after the first 10 minutes, and we had to abort the treatment. No treatment-limiting toxicity was observed. And when I say treatment-limiting toxicity, we define that because of the known side effects related to hyperbaric oxygen, like seizures, pneumothorax, and others, and none of these patients had any of them. So let's look at efficacy. And again, this is interim data, so it's not final results. And this is for the first 12 patients. And here, and the next slide, you're going to see that we match the cohort of the HBO patients to a match group. And these are the groups on the x-axis. And the y-axis is the time to neutrophil recovery on this slide. And you can see here on average, we lower or shorten the period of neutropenia by 0.3 days in the HBO group compared to historic group. For playlists, we do that we shorten that by an average of 3 days. So what we've learned so far. So our experience in terms of hyperbaric oxygen and hematopoietic stem cell transplantation, one, the procedure is very well tolerated. To date, we have treated 35 patients in both of these trials. Also, it looks like there is a hint that we might be getting improvement in terms of neutrophil and platelet count recovery, at least based on our historic data. Very excited about these results. We're going to move it to the next level, which is a phase 2 trial, to further explore the effects on engraftment. Have we learned anything else? And actually we did. So I'm going to bring you back to the idea of the EPO rebound. So we did see EPO rebound after 24 hours. So now I'm back focused in the lab to see if we can alter the schedule of the HBO, maybe timing, maybe frequency. Can we lower EPO a little bit longer to kind of last throughout the period of homing, which is the first 72 hours? And the other question is, if we end up doing that, can we get better engraftment? We don't know, but these are things that we've learned. So I'm going to leave you with a few things. First, I introduced to you a new concept today, which is looking at EPO as a target in hematopoietic transplantation. The other thing I introduced today is the approach of using hyperbaric oxygen, which is novel in the sense that we're not manipulating the graft. We're working with the human microenvironment in favor of engraftment. But what I really learned is that we just scratched the tip of the iceberg. And there is actually more to learn about this, about the biology, about the timing of the intervention, and what other correlatives that we can learn from. I'm going to leave you with this. Stay tuned, because I think in the next few months to a year, or maybe longer, we're going to learn more about this. We have multiple samples that we stored, so we can actually use that to study this. They always say it takes a village, and it does take a village. So this is a short list of who actually helped me with this. I want to point out that the SWOG Hope Foundation grant was an integral part of the success of this research program. I want to also thank the patients, because at the end, they make any effort a worthwhile effort. Thank you.